Hello everyone, welcome to our discussion on sex determination. In this session, we are going to discuss about bar body and lion hypothesis and dosage compensation. So, what is a bar body? So, it was first demonstrated by Murray Barr and Bertman in 1949 in the nerve cells of female cats. So, this particular structures, which are called as bar body, was first reported from the nerve cells of cat, and they were seen as deeply stained bodies which is attached to the inside the nuclear membrane. So in the nuclear membrane, if you stain the cells, or when the nerve cells of the female cats were stained, what was seen is that deeply stained bodies attached to the inner side of the nuclear membrane was observed, and later it was observed that such structures were present in the somatic cells in the females of many mammals. So bar body is also called as sex chromatin because of its association with a particular sex, because this is seen only in females. In especially in our mammalian females, you can see such structures which is called as bar bodies, which is also called as sex chromatin. They're called as sex chromatin because of their association with a particular sex and also their stain loving nature, and the chromatin or the strain loving nature gives them the name. So the bar bodies are observed easily in the epithelial cells of the mouth cavity, the vagina, the urethra of normal females, and this is absent in normal males. So this is usually done to also check whether, or this is a simple test to detect whether that particular individual is a male or a female. So this can prove whether the presence of the sex chromatin or the bar body indicates that the individual is female. So females are said to be sex chromatin positive, and the males are said to be sex chromatin negative, and that is based on the presence and absence of this bar body. So now we are going to see what this actually is. So this is first reported to be a deeply staining structure. So what is that structure which is present inside the nuclear membrane? So this is nothing but it is a DNA, which is deeply stained DNA, which is lying in the inner side of the or the membrane of the associated with the nucleolus. So, what DNA is this actually? So, actually, this structure, which is bar body, is nothing but an inactivated X chromosome. So that we'll come to that in the session, the same session itself. So this particular structure, that is the bar body, is nothing but they are inactivated X chromosome seen in mammalian females. So in diploid cells, the number of bar body will be one less than the number of X chromosome. So in diploid cells, the number of bar body, to calculate the number of bar body, it will be one less than the number of X chromosome inside the cell. So, whatever the condition may be, if a particular organism is having, for say, because of some abnormality, an organism is having three X chromosome. In such cases, what happens is that one X chromosome will remain as X chromosome, and the other two will form this bar body. So, only one X chromosome will be functional in an organism with multiple X chromosome in mammals. So why all the other chromosome except one chromosome? Why all other X chromosomes are getting condensed or inactivated? So this inactivation of X chromosome, which is seen in mammalian females, is as I said, it is the condensed X chromosome, and it is inactive. So condensed structure. Why it is called as inactive? Why you use the term inactive? It is called as inactive because it is seen as a deeply staining structure. Means it is highly condensed because all the DNA or the all the double helical DNA is tightly packed and wound around, as we discussed in the starting of genetics. They are wound around in a solenoid, and it is again wound, and it is tightly and more tightly packed. So it is called as inactive because this DNA or this particular chromosome or the genes of this particular chromosomes is made inactive. They are transcriptionally inactive. They are condensed and condensed so that, and they are also chemically modified. That chemical modification, the chemical modification and the chemical modification means the DNA is not altered. The chemical modification is on the proteins or the histones. The histones are modified in such a way that the histones do not leave the particular complex, so that they are tightly held. When they are tightly held, the particular DNA strand is not transcriptional friendly, or they cannot be transcribed. And because transcription does not happen, or they are transcriptionally inactive. So just inactive means, just when you say a particular chromosome to be inactive, it should be taken to mind that it is transcriptionally inactive. There is no transcription in that particular DNA or that particular chromosome. That's why they are made inactive. So now we are going to see why in mammalian females this particular inactivation of X chromosome should happen. Why one X chromosome should be made inactive. So what happens is, in normal females who are having 2n number of chromosomes, so and 2n number of chromosomes and one bar body. That means in normal females you will have 22 autosomes, one X chromosome, that is two X chromosome will be there. Of that, one will remain as X chromosome and the other will be a bar body. So bar bodies are used to distinguish between normal females and also useful in diagnosing different abnormalities. So abnormalities in the sense, if you have more number of bar bodies, that also can be identified. In the case of an of males who are having a particular syndrome, which is called as XXY. If a particular individual, I'm talking about humans, if an individual is XXY, he will be a male because of the presence of Y chromosome we discussed in the last session. If there is Y chromosome, that individual will have, or that a particular embryo will move towards maleness. And in such individuals, so normally in the case of males, as we said, there should not have any bar body. But if a bar body is present in a male, that indicates that he is having an extra X chromosome, and that is a syndrome which is called as Klinefelter syndrome. So even though that particular individual is having male sex organs, etc., because of the presence of the extra bar body, he will show some inclination towards femaleness. That's why it is called as a syndrome, and mostly they are sterile because of the presence of this condensed X chromosome. They are sterile, and such a syndrome is called as Klinefelter syndrome. So such cases can be identified. You can also identify this with newer technologies. You can also identify that by doing an electron, or an or a SEM image, or an electron micrograph can be done for identifying abnormalities. So other than that, with simple test, with just using a microscope, you can identify this, whether there is some abnormality. So now coming to the question, why the X chromosome should be inactivated in mammalian females. So why? What is the reason why X chromosome should be inactivated and condensed? And one thing to be noted here is that even though the X chromosome is inactivated, we say it is being inactivated and it is transcriptionally inactive. So we said that the X chromosome does not, or not only houses the genes which are related to the sex determination. It also has many genes which is concerned with housekeeping. Housekeeping in the sense, the normal functioning of the body. Like the somatic functions for many genes are present, which also helps in the normal activity of the body, other than sex determination. And this particular condensed X chromosome, even though it is condensed, some housekeeping genes are always active. Even though they are condensed, some genes are still active in them. Some genes are always transcriptionally active in them. And that is why when I described a particular condition which is called as XXY, so in normal cases, what we have to conclude or deduce is that if a particular individual is XXY in that case, we said that whenever there is an extra X chromosome, one will condense and form a bar body. The same thing will happen in that XSY male. In that also, one will form a bar body. So, if bar body is completely inactivated, he should be fine. He should be a normal male. He is not a normal male, he is sterile. And that indicates that that inactive X chromosome is having some genes which are transcriptionally active, which is causing this particular abnormality. So, coming to this question, that is why X chromosome should be inactivated in normal mammalian females. Now, this particular point is about normal mammalian females. Why they are having one normal X chromosome and one as a bar body. And for to explain this, this was explained by Lyon, a British geneticist, formulated a hypothesis which is called as Lyon hypothesis to explain this formation of bar body or X chromosome inactivation. And this is explained by the phenomenon which is called as Lyon hypothesis or dosage compensation. So, to describe this particular hypothesis or dosage compensation in simple words, what nature has done is that if you take the organisms or mammalian or mammals, we are having the sex determination which is XXXY type of sex determination. In that, females are having two complement of X chromosome and males are having only one X chromosome. Y chromosome cannot be considered as equal to X chromosome because it is having only about one tenth or less than one tenth of the genes when you compare it with the X chromosome. So, in that particular, or if you take humans, you can see that males and females, they're 1 to 22 pairs of chromosomes are the same. They're having the same genes with variations, etc. Whatever it is, they are having the same type of chromosome, they're having the same type of genes, etc. But only the difference comes in the sex chromosome, where females are having extra complement, whereas males are having one complement less. So to compensate the dose, so compensate in the sense if it was normal X chromosome, females would have more amount of housekeeping genes or housekeeping proteins or whatever it is, it being produced in females. So nature doesn't want that, and nature what it does is to make the dose equal, the doses of chromosomes equal in both males and females. One of the X chromosome in every cell in the female's body will be every cell in the sense all the cells, all the somatic cells except the ogonial cells. In the ogonial cells or in the ovary, the eggs or the cells which are forming the ova, in that only in those cells this both the X chromosomes are active because an egg is being formed. Other than that, all the cells in the female's body will have an X chromosome which is inactivated and condensed so that the dose of X chromosome in males and females are the same. And this is what is called as dosage compensation. So the reason why the X chromosome is inactivated is that it is to equalize the dose of genes present in males and females. If both the X chromosomes were completely active, females would have had double the number of a particular set. There will be double the number of a particular set of genes in females when compared to males. So, to avoid this, there is something, the process called as dosage compensation, which is happening. So, dosage compensation can happen in many ways. In mammalian females, it happens by the formation of bar body, where an X chromosome is inactivated and contains. So, coming to this, in organism with the sex determination, which is XY, the Y chromosome is a degenerate one, and the X chromosome is the one which there are very few functional genes other than that concerned with sex determination itself, concerned with sex determination in sperm production or in both. The X chromosome, on the other hand, contains many genes concerned with basic cellular processes, which are called as housekeeping genes, which are always active. In species with XY sex determination, The females have two X chromosomes, whereas the males have only one. So even though there is two X chromosomes in females, the X chromosomes housekeeping genes are expressed in equal intents or equal amounts per cell in females and in males. So even though there are two X chromosomes, it can be seen that the amount of the genes or the gene product that is produced by the housekeeping gene from the X chromosome is almost equal in males and females, and that is or that is brought about by the process, or in other words, there is a process called as dosage compensation to accomplish this particular task. To accomplish this, that is to accomplish the approximately equal extents of transcription from the X chromosome in both male and female, you have the process called as, or the phenomenon which is called as dosage compensation. So to achieve dosage compensation, what the X chromosome does is one of the X chromosome in the female cells is chemically modified. That's why I said chemical modification does not mean that it is the modification of the genes. Genes are not modified. If the genes are modified, it gets mutated. So genes are not modified. It is just the modification of the proteins or the histones so that they are or they remain tightly closed without any without or they do not move out without that chemical modification is removed and condensed into very small mass. And as I said earlier, X chromosome inactivation is the process of, so dosage compensation is the term dosage compensation just means that the organisms which are having XY type of sex determination, XXXY type of sex determination, to compensate the dose in males and females, one of the or some mechanism should happen, and that mechanism is called as dosage compensation. And dosage compensation in mammals and especially in humans, that is done by inactivation of the X chromosome. But in Drosophila, there is another phenomenon or another mechanism for dosage compensation, and that is what we are going to say now. So, in fruit flies or Drosophila, the males are having X chromosomes appear to be hyperactivated. That can also be done to equalize the dose. That is, females are having two, males are having one. So, what happens in Drosophila is that the X chromosome in male Drosophila is hyperactivated. That means it produces double the dose of transcripts or the gene products from the X chromosome in male. That can also be done for that. Also leads to what is called as dosage compensation. Our intention is to compensate the dose or equalize the dose in males and females. It cannot be always as happens in mammals by inactivation. This is also a mechanism of dosage compensation. So here dosage compensation in fruit fly is by hyperactivating the X chromosome. That means the X chromosomes are more activated. That is the one X chromosome in the males are hyperactivated, allowing it to be transcribed twice at the rate other either than the or the of rate of either X chromosome in the female. As a result, the XY Drosophila has an X gene dosage equivalent to the XX female. So, this is a mechanism by which the dose is compensated. So, dosage compensation uh, can be by hyperactivating a chromosome and also by inactivating a chromosome. So, the second one is going to be discussed now. So, in mammals, in contrast to this rule, no matter how many X chromosomes are present, there is only one transcriptionally active X chromosome in each somatic cell. So this is a simple uh, or a one-line definition of what happens in mammals. That is, the rule is that no matter how many X chromosomes are present, there is only one transcriptionally active X chromosome in the somatic cells. So that is also to be noted. Somatic cells. So only in somatic cells you see this dosage compensation, not in the germ cells. In germ cells of every mammalian females, both the X chromosomes are active. So now a question arises. If the or if in the oogonial cells or in the germplasm or whatever called as the germ cells or the ova or the oocyte, if the X chromosomes are active when is it inactivated? So when you observe a female, or when you are analyzing a female, you can see all her somatic cells are having this X inactivation, and only in the oogonial cells, or the oocytes, or the cells which are forming the ova and the oocyte or the egg, you can see it is active. And now the question is when it is getting inactivated. So the answer lies here. So this rule gives the XX female mammal and X gene dosage equivalent to that of the male. So dosage compensation and achieved is by X chromosome inactivation. So a female with two X chromosomes, for example, is a mosaic with two types of cells in which one or the other X chromosome is active. So here comes the answer for our question, that is when and how this inactivation occurs. So a female is said to be have two X chromosome and is a mosaic of two cell types in which one or the other X chromosome is active. So what is this one or the other X chromosome is active? Okay, so this before explaining this, we'll see this and come to that particular point. So now our question was when it is getting inactivated, and the answer is the X chromosome inactivation occurs at an early stage of development. Early stage of development in the sense that when an oocyte is formed, or the oogonial cells, as I said, there is no inactivation because in the oogonial cells, the cells which are forming the oogonia, or when they form the oogonia, the inactivation process is reversed. They become the normal X chromosome. And what happens is, and the oocyte is formed, the egg or the ovulation happens, the egg is released, it meets a sperm, which is called as fertilization, a zygote is formed. As zygote starts cleavage, it starts to divide, and the process of X chromosome inactivation occurs at an early stage of development and is a random process. That is, any of the two X chromosomes can be inactivated. Okay, and this inactive state will be propagated to all the progeny of cells, and this inactivated chromosome is called as the Barr body. So now we are going to see how this inactivation process happens and how this is being propagated. So these are the terms which are going to be concentrating now: the propagation and the mosaic. So how this inactivation is propagated, and why females are called as mosaic of two types of cells with regard to the X chromosome. So what we are going to see here is, so I'm going to take a zygote. So this is a zygote where both the X chromosomes are active now. So in this, both the X chromosomes are active. So both the X chromosome, in the sense that one of the X chromosome is from. So if this consider this as a zygote. So this is a zygote. This one is a zygote with a nucleus inside, with a diploid nucleus inside, and this particular zygote is having two X chromosomes. It is a female, so that's why it is XX. One of the X chromosome is from the father. The father, from the sperm, you get one of the X chromosome. This is of the father, and this X is contributed by the mother. Okay, so you have two X chromosome. So now the question is, which X chromosome should be inactivated? Fathers or mothers? So that is the question, and that is what is called as, or what we are going to see in propagation and mosaic. So what happens is, after a zygote is formed, which is a one cell stage, now this starts to divide. From one cell, it cleavage starts. It's formed two cells. Okay, and from this two cells, again it will divide. It will form four cells. Okay, so in this particular four cell stage, for example, we are considering it happens either in the four cell stage or after four cell stage, 8 cell or 16 cell stage. This particular X chromosome inactivation occurs. So now we are considering this particular X chromosome inactivation is happening at the stage which is called as a four cell stage. Now the embryo is having four cells, and each cell will have two X chromosome. Okay, no doubt, because mitosis is happening. There will be two X chromosomes each, plus the autosomes are there. We are not going to discuss that now. We are going to discuss only about the X chromosome. All the autosomes are present. Consider all the autosomes are present in all the four cells. This is a normal female with two X chromosomes, and the two X chromosomes comes from one from the father and one from the mother. So what happens is, we are taking this four cell stage because to make our discussion easy. Uh, Actually, it happens after the four cell stage, either in the 8 cell or the 16 cell stage. This inactivation happens. So what happens? All the four cells are having four cells, which is shown here. Four cells of the embryo, which is also called as a blastomeres. They are having two X chromosomes each, because that is being going to be developed into a female organism. This is a four cell stage of an embryo. So in this four cell stage, what happens is that the X chromosome inactivation, as I said, which X chromosome should be inactivated? Should it be fathers or should it be mothers? Okay. And what decides whether fathers should be inactivated and mothers should be inactivated, or whether in the case, or all the mother's chromosomes are inactivated or all father's X chromosomes are inactivated? So what happens here, or what is a deciding factor here for which of the X chromosome in each cell? Every cell is distinct, and this event should happen independently in every cell. And this particular process is said to be a, as noted here, this is a random process. Random in the sense that it does not depend on the other cell. That means, if we take an example, in this four cell stage, in this particular cell, for example, the father's X chromosome is getting inactivated and the mothers are normal. In this particular one, for say, the mothers might be getting inactivated. In this one, again, mother's X chromosomes might be getting inactivated. And in this one, again, sometimes the mothers might be getting inactivated. It does not mean that because I have drawn three inactivation of mother and one inactivation of father, does not mean that every time this happens. This is a random event. To show this randomness, if I draw two fathers X chromosome and other one, it will be 50-50. It is not 50-50. It is a random event. It can happen anyways. So a random inactivation of X chromosome happens. So in one cell it might be mother's, the other cell it might be the same, it might be different, etc. That's why I took it just as a random. Okay. So here this is the process of X chromosome inactivation in the zygote, or in the early stage of development where this inactivation happens, is a random event. And after this random event, what happens? Because again, these cells are going to divide. These cells again, the four cells again divides by mitosis, produces 16 cells, 8 cells, 8 will produce 16, 16 to 32, 32 to 64, and goes. And at last, a complete uh, the embryo or a complete female will be formed. So in this particular case, when a complete female is formed, in this particular random process or a random manner of inactivation occurs. And here you can see that the next term that we are going to discuss is the after random, this propagation. Okay, so you have a random process, and after the random process, this inactive state will be propagated. And propagation is in the sense that, if for example I am going to take the example of this particular cell, I take this particular cell where father's uh, X chromosome is activated, and please assume that this particular cell, it will divide, it will form two cells, four cells, etc. And this is going to be part of the head of that particular, or this is going to be forming the part of the skin, or the upper part of the skin of upper part of the skin, or the skin in the case of the this cell will be part or dividing, and it will form the part of the skin in the female. It forms the part of the ectoderm or the skin. And in that case, the female skin will be having the X chromosome, or the skin cells will be having father's chromosome, which is inactivated. So, as you all know, skin is having two layers. One is epidermis. So, I talked about the epidermis. So, the first cell, which is shown here, it divides and forms the layers of the epidermal cells, or it is the precursor of the epidermal cells. So, all the epidermal cells are in a female. In this particular female is having its father's X chromosome, which is inactivated, and you have an underlying dermis. And consider this particular cell, that is the cell which is here. So where is it? So consider this particular cell. This is forming the dermis. So if this is forming the dermal region, which is underneath the epidermis, if this forms the dermis, in that dermis you can see that it is not the father's X chromosome, it is mother's X chromosome is being inactivated. And this is why you call a female with two X chromosomes as a mosaic. A mosaic in the sense that some of her cells, or it might be random, in some of her cells, in some of the cells the X chromosome of the father will be inactivated, in the other cells X chromosome of the mother will be inactivated because of this random event which is happening early in development for X chromosome inactivation. So a female with two X chromosomes is a mosaic of uh, two cell types in which one or the other X chromosome is active. The X chromosome inactivation occurs at an early stage of development and is a random process. That random process is what we discuss now, and that means any of the two X chromosomes can be inactivated. And after the inactivation, this inactive state will be propagated. That is, if one in one cell maternal is inactivated, the progeny of those cells will be maternal inactivation. In another cell, if paternal X chromosome is inactivated, the progeny of those cells will be having paternal inactivation. So that is why the inactive state will be propagated to all the progeny of the cell, and this inactivated chromosome is referred to as the Barr body. So this is our discussion of we discussed about the Barr body. So Barr body is a condensed chromosome or the condensed X chromosome that is usually seen in mammalian females, and that is a structure which is due to the phenomenon which is called as dosage compensation. And dosage compensation is to equalize the dose of the X chromosomal genes in males as well as females, and that is done by inactivating or condensing the X chromosome. And this condensation is seen in every female cell except the gametic cells. And now the question, when this inactivation occurs, this occurs at an early stage of development, and this process is random. It happens, inactivation happens randomly in paternal and maternal X chromosome. As a result, in a female body you can see both type of cells. That means the cells with paternal X inactivation and the cells with maternal X inactivation. As a result, with regard to the X chromosome inactivation, mammalian females can be called as mosaics, and this inactivated state will be propagated. That is, once inactivated during the early stage of development, that inactivated X chromosome will be propagated to the next, or the cell lineages which is formed from that particular inactivated cell. With this, we come to the end of this discussion. So, in the next session, we'll be discussing the third type of sex determination, that is a hormonal influence of sex determination. Thanks for hearing.